In the 14th century, the Black Death killed about 30% of Europe's total population. But thanks to the discovery of antibiotics, we have been protected for almost a century. Unfortunately, that might all change with the rise of antibiotic-resistant superbugs. But there is hope that AI will help us find new antibiotics. In this paper, the researchers used AI to find Abaucin, a new narrow-spectrum antibiotic. The paper was published in May of 2023 in Nature Chemical Biology, and the title is Deep Learning Guided Discovery of an Antibiotic Targeting Acinetobacter baumannii. Here on the right, I have a little outline of the paper to help me guide you through all the steps they did to get to the final compound. First, they screened 7,500 molecules in vitro for activity against this bacterium. So that's shown in figure 1b right here. So you can see they screen over 7,000 compounds, and the ones signified in red are considered to be active because they inhibited the growth of bacterium in the culture. So they did this in plate cultures, and anything that showed slower growth was considered to be active. So this was their data set. Their cutoff to be included in the data set as an active molecule was 20% inhibition of the growth. Then they used this data to train their neural model. So that's signified here in figure 1a. So the training data comes in here through this funnel. So that's the training data from this figure. And then they train the model. Here's a little bit of information about the model. So this is in the method section under model training and predictions. A directed message passing neural network, chemprop, like other message passing neural networks, learns to predict molecular properties directly from the graph structure of the molecules. If you're not familiar with chemistry, it means that the model is learning directly from the molecular graph, which is just a graphical representation of the molecule. The model applies a series of message passing steps where it aggregates information from neighboring atoms and bonds to build a representation of local chemistry. If you want to learn more details about the model, it's really nicely explained in the method section, and also the code, the chemprop, is available on GitHub. So once they trained the model, they applied this model to prediction set compounds. This was another 6,680 molecules that have been not included in the original training data set, and they applied this model to this new set of compounds. What I like to highlight is in the outline that only these two yellow steps are where AI was involved. Everything else in this paper is just biology. If you're finding this content useful, please consider subscribing. So once they applied this model to the 6,680 molecules, the model spit out 240 priority molecules. So that's highlighted here in figure 1c, where you have the molecule rank. So this is the 6,600 molecules and prediction score, and the cutoff they chose was 0.2. So any molecule that had a higher score than that was included for further consideration. And that resulted in 240 priority molecules. So they reduced the set of molecules by 96.4%, which is really good. But it's not as high if you consider that in the future, they might be screening millions or maybe even billions possible molecules. Then they purchased these 240 priority molecules, and then they tested them again in vitro for antibacterial activity. Here they chose a higher cutoff of 80% inhibition cutoff, and that resulted in 9 molecules. If they use a less stringent cutoff of 20%, then that would result in 41 molecules. The 9 priority molecules were subsequently assessed to remove 1, those with major structural features that observed in known antibiotics, so they wanted to remove any molecules that are already known to be antibiotics. 2, those with reported antibacterial activity from the scientific or patent literature, so again, molecules that are known to be antibiotic. And 3, those with possible non-specific membrane activity as assessed by the presence of acylic aliphatic moieties. After that, they ended up with 2 molecules, RS102859 and serdenmetan. The first one was more potent, so they chose to study that one further. The RS102859 is a well-studied CCR2 selective chemokine receptor antagonist, but the antibiotic property of this compound is new, and that's the true contribution of this paper. And this antibiotic property of this molecule was in large part found with using AI. For its antibiotic properties, they renamed RS102895 Abaucin for its activity against Baumannii. Overall, I think this is another big step forward for biology, and it shows you the potential power of AI in the drug discovery process. Thank you for watching. I'll see you next video.